Welcome to the One Health Seminar Series. My name is Catherine Hayland. I'm a Program Manager for the One Health Institute, and it is my pleasure to be facilitating this seminar today. Before we begin, I would like to acknowledge with great respect that the University of Guelph resides on the ancestral lands of the Attawandaron people and the treaty lands and territory of the Mississaugas of the Credit. We uphold the significance of the Dish with One Spoon Covenant and the continuing relationship our Indigenous neighbors have with this land. We recognize that this gathering place and all the places we are joining from today is today home to many First Nations, Métis, and Inuit peoples, and acknowledging them reminds us of our relationships to this land where we explore, share, discuss, and learn about our shared responsibility for human, animal, plant, and ecosystem health. Before I introduce our speaker today, I wish to inform you that we are recording this seminar. The recording will be posted on the One Health YouTube channel, and a link will be made available on the One Health Institute website. The recording may also be used by the One Health Institute in the context of promoting and showcasing One Health work. It is now my pleasure to introduce Dr. Whitney Preisser from Kennesaw State University in Georgia. Dr. Preisser is an assistant professor of disease ecology. Her research program looks at how the environment, hosts, and anthropogenic disturbances shape spatial and temporal distributions of parasitic diseases. Dr. Preisser received her PhD from Texas A&M University in 2019 and was a postdoctoral scholar in the School of Aquatic and Fishery Sciences at the University of Washington from 2019 to 2021. She was then a biodiversity postdoctoral fellow in the Department of Organismic and Evolutionary Biology and the Museum of Comparative Zoology at Harvard University from 2021 to 2022. I'm very happy to be able to turn the mic over to Dr. Preisser.
A parasite is an organism that lives in or on another organism, and it gets its food from or at the expense of that host. So it uses a host organism for resources, and by definition, the parasite causes some level of harm to their host. It's often physiological and or mechanical harm. So here we have the stomach lining from a rodent, and you'll see squiggles inside the stomach lining. Those are from parasitic nematodes or roundworms. And so in this case, the roundworm is causing some mechanical damage to the lining of the stomach just by being there, just by its presence. Because parasites can consume the host nutrients and resources, they can take those nutrients and make them unavailable for the host, so the parasites are causing harm in that way. And they can also take up a lot of space. So here we have a stomach from a rodent, I think it was in Nebraska, and you see it's full of worms. And so because of this, the rodent, when it eats, it can't fill up its stomach as much as it would normally do, and so it may have to eat more frequently and larger amounts of food in order to make up for what the parasite is taking. So in that case, it takes harm or causes harm to the host. Parasites can also alter the behavior of their host to further their life cycle, which can result in host damage or even host death. So this is a nematomorph. It's a type of parasite that usually uses invertebrate hosts. And so they live inside the invertebrate host until they mature, and then they will burst out of the host. They will cause their invertebrate host to jump into the water, and then they burst out. So they're causing mechanical damage because they cause pressure necrosis. They're causing a major wound whenever they burst out. And then sometimes the host drowns in the water after it jumps in. So again, we have some harm to the host. Another example, I'll see if this video will play, is a trematode larvae that lives inside the eye stalks of a snail, and they are causing the snail to spend more time out in the open, so it's more likely to be eaten by a bird, because this parasite wants to move into a bird where it can reproduce. And so it causes harm to this snail. It changes the behavior, and it makes it more likely to be eaten by a bird. And then lastly, some parasites can reduce or stop host reproduction. One example of this is through castration. So this benefits the parasite because the host is no longer devoting energy towards reproduction, and instead that energy can be used for the parasite. It's often said that the host is now no longer its own animal because it can't, it has no fitness anymore. It's an extension of the parasite. All that energy goes towards the parasite and not towards reproduction anymore. So obviously that is harmful to the host. And the parasites can also reduce a host immune function, which can make them susceptible to other diseases. So animals mount different types of immune responses depending on the threat. And so if a host is mounting an immune response towards a parasite, it may be more susceptible to other diseases, whether they're bacterial, viral, some other parasites. So to an individual host, parasites are harmful. The level of harm can range anything from an annoyance to death, but it causes harm in some way. However, I'm going to argue that outside of the harm to an individual host, parasites are actually really important. And I want to preface this by saying I'm not talking about human parasites. Human parasites make up about 4% or less of all parasites. They are harmful to people. We should not conserve them or protect them. They should be eliminated. And domestic animal parasites are also problematic. But in wildlife, parasites are supposed to be there, and they play really integral roles in ecosystems. So they can help control population dynamics and help control population sizes. Here on the right, we have a graph of a boom and bust cycle for red grouse, which is a type of bird. And the parasites cause population crashes, and then the grouse recover. Parasite causes more population crashes. And we know it's from the parasite because when they're treated, which is what those stars are, when they're treated for parasites, we see that the crashes go away. But I would argue the population crashes are actually important for the red grouse because it helps prevent them from getting too numerous and starving or outcompeting other animals. So parasites are really important for many population dynamics. They also help move energy through ecosystems. So on the right is a food web with just the free living species in red, and then our primary producers in green. And the food web on the left includes the parasite, which are the blue spheres. So parasites infect animals at every trophic levels, and many parasites are trophically transmitted. And so they're able to help move energy through an ecosystem. And they also serve as prey. And then also, as I mentioned earlier, parasites can also influence energy allocation within a host. And another way that parasites are important for ecosystems is through predator-prey interactions. So like the behavioral manipulation I talked about earlier with the nematomorphs, parasites can increase the likelihood of prey getting eaten by their predators. So they can do this either by making them weak, more likely to be out in the open. There are multiple ways that they do this. But they often do it to complete their life cycle. So the nematomorphs want the crickets to jump into the water so they can then burst out of the cricket and continue their life cycle. But the crickets can serve as really important food sources for aquatic animals. And in this one study, they found that 60% of the trout's diet was made up of these crickets. So the parasites are responsible for essentially feeding these fish. So overall, wildlife parasites are really important parts of natural ecosystems. And it's been argued that healthy ecosystems are full of parasites. But if they're so important, why do we know so little? Because we know very little about parasites. And I think there's a few reasons for this. First, they're really small, and it's really to say out of sight, out of mind. If you're not looking for them, it can be easy to ignore them, even though obviously I don't think that's right. They're also not easy to find and collect in a lot of cases. Studying parasites can require killing other animals sometimes, and this isn't easy or feasible or recommended for many host species. And then lastly, I think because they've been ignored for so long, it's easy for people to continue to ignore them. It's a really cyclical a really cyclical problem. But if we ignore parasites in ecology or really any field, we're ignoring a major part of the biodiversity on Earth, as it's been estimated that 40% of all life on Earth is parasitic. So we're really doing ourselves a major disservice. Now, oftentimes when people go to study animals, they turn to natural history collections. And parasites are in natural history collections, but unfortunately, they're really not well represented. There are about as many bird specimens in museums in the US alone as there are parasites in any museum in the world. And there are so many more parasite species than there are bird species. And so because they're missing from museums, it makes studying long-term changes very difficult. So normally we would turn to museums to use older specimens so we can look at long-term changes to populations, to species, looking at how they're affected by different, environmental or human factors. But we can't really do that for parasites because they really don't exist in collections. They're very underrepresented. And so it makes it really difficult to study, changes in disease risk or monitoring populations or managing species. It makes it really difficult because they're not really there. And so where do we get this information? Well, actually, fortunately, there are millions more parasites hidden in the collections if you know where to look. So depending on how they were prepared, many dry specimens will still have some of their ectoparasites, especially those with low vigility or those that can't move very well, like lice. And then fluid preserved specimens are even more valuable because they can hold the parasites that were infecting them at the time of death. So essentially, these animals serve as time capsules, and we're able to use those older specimens, open them up, and find the parasites that were infecting them at the time of death. Now, this is really, really invaluable because we can get an almost complete picture of what parasites they had at the time. And this is, major advantages over museum specimens because there's a lot more complete specimens. All the parasites are there. And so it's a really, really great resource to be able to look into the past and look how parasites are changing through time and in response to what factors. So today I'm going to tell you about a few projects that I worked on previously or are currently working on that use this really valuable method to examine the historical ecology of parasites. So I was first introduced to historical ecology when I was a postdoc at the University of Washington. And we wanted to look to see how humans were impacting parasites. So we know that human influence is threatening many aquatic and terrestrial species, but we don't really know what is happening with their parasites. And we don't know how things like climate change or pollution or overfishing are impacting the parasites. And so we didn't come up with this method. It's been done a few times elsewhere, but we really took advantage of it. And we use these museum specimens, open them up for parasites, and can get this really, really valuable data that we otherwise wouldn't have. So first we needed to validate the method, make sure it would work. So we sampled English sole, that's what's pictured here, from the museums. These sole were collected in the Salish Sea, which is in Washington State, which is in the northwestern U.S. We sampled fish that were collected between the 1930s and 2019, and we aimed for about 10 to 15 fish per decade. And I just want to put a little shameless plug in here. If you are interested in working with fluid preserved specimens for parasites, we have this really cool paper that we wrote up that details exactly how we did it and recommendations if you want to do it yourself too. But back to our study, pictured are some of the parasites that we collected from these fish. They're not quite as nice as fresh specimens, but for being almost a century old, these parasites are in great shape. So it really allowed us to ask some cool questions about parasite communities. So the first question we wanted to ask is, are parasites declining through time? Like we see many fish declining over time from overfishing, pollution, etc. What's happening to the parasites? And we expected they were declining over time as well. But surprisingly, when we looked at the number of parasite species in each fish from 1930s to 2019, we actually found no change. And this is incredibly surprising to us because the area that we sampled from has seen intense industrialization, urbanization. There's been a lot of over-harvesting from fish. And that given that, fish are declining, we expected to see the same thing in parasites. So this was really, really surprising to us. Okay, so we thought maybe if the number of species isn't changing over time, maybe infection burdens are. So, maybe fish are more wormy now than they were then. And so taking that data, all the species that we had that were in more than 5% of fish, so we had a good statistical power, we tracked how they changed over time. And so of the 12 parasite species that were in more than 5% of the fish, surprisingly, again, I'm going to use this word a lot, we're surprised by a lot of our results, only 3 species significantly changed over time. So of these 3 species that changed, 2 decreased in abundance, so they became less common in fish, and 1 increased, it became more common. And the different directions could be caused by multiple things. So all 3 of these parasites have what are called complex life cycles, meaning they use one or more hosts to complete their life cycle. They have at least one host where they do development, and then they have a definitive host where they reach adulthood and they reproduce. And so with complex life cycle parasites, if something goes wrong with just one host in their life cycle, it can cause major problems for the parasites. So it's possible that one of the fish or invertebrates or other animals in the life cycle of the 2 species that declined, it's likely that one of those host species declined. And so then we're likely to see a decrease in the parasite as well. They're a little more fragile, these complex life cycle parasites, because if one thing goes wrong in the life cycle, the parasite's affected everywhere. For the species that increased, it might be increasing because it uses a host that is increasing over time. So this is something we wanted to look into more detail with and sort of figure out, why are these changing? Now, so we could use the English sole data to look and try to correlate and figure out, are humans impacting these populations? Is that why we're seeing decreases or increases? But it was only 109 fish, which isn't very many when you're looking over the, 100 or 89-year time span. So we made this study a lot larger. We collected over 17,000 parasites from about 700 fish specimens from these 8 species pictured here. And we're able to increase our temporal span as well. So we looked all the way back from the 1880s, we had some really, really old fish, all the way to 2019. And these fish have a variety of trophic levels, diets, habitats, depths, to give us a really good picture of a lot of the parasites in the system. So we collected parasite data on all of these fish, and then we were able to cobble together from multiple sources data on sea surface temperature to represent climate change, heavy metal and organic pollutants, and then we were able to get some measures of fish abundance for some species as well. Because we wanted to pair these human disturbance data with the parasite data to see if we can find some patterns. So unlike our study with the English sole, we saw a lot more than 3 species significantly change in abundance over time. So each parasite is listed on the left. They're in code, so don't try to read them. If you're interested in the codes and what specific parasites changed, that's in our paper, but I just want to talk about broader patterns here. So the line down the middle is 0. If a little parasite horizontal line overlaps with 0, that means there is no significant change over time. If it's to the right of the line, it means it's significantly increased over time. If it's to the left of the line, that means that it's significantly decreased over time. And I made it easier. I labeled with red arrows all the species that change significantly over time. And I want you to notice a few patterns here. The majority of parasite species are decreasing over time. We are seeing their numbers fall between the 1880s and 2019. And I also want you to notice I have this split by the number of hosts that each parasite uses. So the parasites at the top in the yellow, they use just one host. The parasites in the green use 2 hosts for the life cycle. And the parasites that are in blue at the bottom, they use 3 or 4 hosts in order to complete their life cycle. And you'll notice that the parasites that use 2 or more hosts are more likely to decline. There's a larger proportion of those species declining than the parasites that use 1 host. And again, the parasites that have complex life cycles, that use more than 2 hosts, there are more places for that life cycle to go wrong. If just one of the hosts in their life cycle declines, we are likely to see a corresponding decline in parasites. So these complex life cycle parasites are more fragile. And I also want to point out, as an aside, 2 nematode species that belong to genera that infect humans. They both are anisakids. They're in the family Anisakidae. And these parasites have been shown to infect people. And they are both declining over time. So overall, we found parasites that use 3 or more hosts, which was a group that comprised 52% of all of our parasites. We found that they declined in abundance at a rate of 10.9% per decade. Now that's actually a crazy high number. If you compare it to other studies, we put it in context, we see that parasites are declining at a rate faster than North American birds, faster than terrestrial vertebrates, and faster than terrestrial insects. Now, when the bird, vertebrate, and insect studies were published, there was a lot of panic, a lot of news articles, a lot of attention paid to those declines because, yes, they're bad. But parasites have a larger decline and nothing. There's very little attention. Most people don't care, which is bad. Parasites are very important, as I explained earlier. So if we take a closer look at the species that decrease, each of these boxes represents one parasite species. And we looked at their presence or absence over the sampling period, and we find that actually 10 species disappeared from our sampling. Now, because of our sample sizes, we weren't able to tell if they were truly extirpated from the system or if they just declined in abundance and became more rare, or maybe we just weren't sampling the right individuals. We can't really tell, but really, rare versus extirpated, neither of those options are good. And I also want to call your attention to the colors again. So remember, yellow is parasites that use one host, and the blue are parasites that use multiple hosts. And so again, we see the parasites that use multiple hosts are more likely to disappear from the system, from this ecosystem, than the parasites that use one host. So now let's turn to the potential anthropogenic drivers, so human influences on this system. We looked at heavy metal and organic pollutants, and we also looked at overfishing or a fishing abundance over time. Neither of those were significantly correlated with parasite abundance. However, when we looked at sea surface temperature, we did find a significant correlation between parasite abundance on the y-axis and sea surface temperature on the x-axis. And when we look at parasites that use 3 or more hosts, so those complex life cycle parasites, we find that their abundance is decreasing at a rate of 38% for every degree Celsius increase. That is a lot. And while these results are specific to the Salish Sea, in Washington State and the northwestern U.S., we do expect at least possibly similar trends for other marine parasites and potentially other aquatic parasites as well. Climate change is a major threat to free-living species, and so we would expect it to threaten their parasites as well. And that's what we found. So in conclusion, we found that different parasite species respond to these external pressures, whether it's natural variation or different environmental variables, species interactions. The species are doing different things. Some species are declining, some are increasing, some have no change at all. But it's really important that we tease those species apart and we look at complex life cycle versus single host life cycle parasites because they respond differently. And if you group them together, you might obscure those patterns. And again, it seems like climate change is a major threat. Now, it's possible that our study was correlative, so we can't definitively determine causation. And it's possible that some other variables are varying with sea surface temperature in the same direction that are actually affecting our parasites and not climate change. And that's something, we will do more work on and try to figure that out. But it does seem likely that sea surface temperature would affect parasites. We know from some experimental studies that parasites have a thermal tolerance range, just like most every other species. And then when you go outside that range, it can cause parasites to die. Now, many parasites also, when they're in warmer conditions, they speed up maturation, they change the timing of their reproduction. And these changes in the timing of things can make a mismatch between when their hosts are available and when they're actually looking for hosts. And so because of that, if the mismatch is there, we would also expect parasite abundance to decline. And so there is some experimental work that does back up climate change or increasing sea surface temperature as a threat to parasites. But more work needs to be done to determine, how universal this is. And so along those lines, I want to talk a little bit about my current work because I did all that work as a postdoc. But since 2022, I've been an assistant professor at KSU, which is outside of Atlanta, and I'll have a map in the next slide. And I'm very fortunate to be able to continue very similar work here. And I have some amazing undergraduate and graduate students who have been able to, really push my research forward. And so on the right, we have a map of the US and Georgia, and then I also put a red box around where we're located in the metro Atlanta area. Now, KSU has our very own fish collection. It's pretty small, about 2,000 specimens, but it's a really great resource for us. There's about 120 fish species in this collection, all freshwater. They were collected between 1999 and 2016 in Georgia. And this is actually a really great location to look at human influence on parasites. For one, the southeastern U.S. is a hotspot for freshwater fish diversity, and that means also it's a hotspot for their parasites. And then two, the metro Atlanta area is one of the fastest growing metro areas in the U.S. We've experienced major population growth over the past few decades, and that's expected to continue until at least 2050, if not beyond. And so because freshwater parasites and freshwater fish parasites are understudied, it really puts me in a good position to be able to fill in major gaps in our knowledge about what parasites are here, how they respond to the fish biodiversity, and then how they respond to human influence in this very urbanized area. We're still in the midst of data collection, so a lot of my results today are going to be preliminary, but I'm very excited to see what these projects turn out to be. So I'm going to talk about some preliminary results from two projects which are spearheaded by my graduate students. So of those 2,000 fish species or fish individuals, we've dissected about 600 fish individuals so far, which it almost puts us as many as that previous study. So I think we're doing pretty well. I don't have an exact count of the parasites as we're still going through and IDing them, but if I had to guess, it'd be between 8,000 to 10,000 parasites that we've found so far. Now, using almost all of the specimens we've dissected, my grad student, Nick, is looking at 3 main localities around a dam here in Georgia. So previous work has found that damming can affect fish populations, invertebrate populations. It can reduce them. It can extirpate them from the system entirely. And usually it also homogenizes the community, so it reduces the number of species of invertebrates and fish around a dam. And so we were interested to see, is the dam doing the same thing for parasites? And then in addition, these areas are also nicely situated for us to look at how urbanization impacts parasites as well. So we'll include some measures of human disturbance in our analyses too. So our first question, similar to my previous work, how have diversity and abundance of parasites changed over time? And so this is some preliminary results using about half the fish we've dissected so far. We have year on the x-axis, and we have either total richness or abundance, which you transform just to make it on the same graph, on the y-axis. And we find that richness and abundance of parasites are significantly declining over time. Again, we did the study from about 1999 to 2016. And so we are seeing parasite populations decrease. Now, we wanted to look at some potential, human influences. How are humans impacting parasite populations? So we wanted to pull some anthropogenic disturbance data. And so using multiple resources, we chose crop coverage and impervious surface as 2 of our measures. Again, this is preliminary data. We're adding more. But we chose crop coverage because when you plant crops, especially on larger scales, you use a lot of insecticides, pesticides, and fertilizers. All of these have been shown to get washed into aquatic systems, and it can impact aquatic systems in multiple ways. So fertilizers can cause blooms of algae and invertebrates. Pesticides and insecticides can negatively harm invertebrates and the fish and the parasites. So this was, I think, a good starting place for us to look at human influence. And then we also chose impervious surface area. So this refers to like parking lots and roads where there's, impervious, the water washes off the roads. And so it carries chemicals like salt, oil, gas. It carries microplastics from the tires, and all that gets washed into the system. And so impervious surface area is also a good measure for human influence in an area. Now, when we looked at both of these variables and how they might potentially correlate with parasite richness and abundance, we found what we expected. Increasing crop coverage, so more crops and more impervious surfaces, is correlated with fewer parasites, both richness and abundance. So it does seem that human influence might be causing parasite populations to decline. Now, I mentioned microplastics, and I want to circle back to that because my other student, Jade, is looking at how microplastics and parasites might interact within a fish, how they might affect each other. So just a brief explainer, microplastics, I'm sure people are familiar with them. They've been in the news a lot lately. They can cause a lot of problems in people, and they cause a lot of problems in animals too. And microplastics can come from a variety of sources. So they come, when you wash your clothes, they can get washed out in the water. Again, tires are a major source of microplastic, and that gets washed into the water. Litter, discarded gear, many, many ways for microplastics to get into the water. And there have been actually a good amount of research on how microplastics are infecting vertebrates. And so in fish, we see that microplastics can decrease survival, it can cause immunodepression, it can reduce reproduction, it can mess with the endocrine system, it can cause physical damage. And in particular, we think that the reduced immune function, reduced antioxidant capacity, and reduced damage to the damage to the gills and intestines might be affecting parasites. There's been very little work done on parasites and microplastics in general. It's a very, very new field. And most of the studies that have been done are using experimental studies. So in the lab, they dose fish with parasites. They dose fish with parasites and microplastics and see what happens. But there's some very little work done in wild systems. So that's what we want to look at. Because microplastics can affect these things in fish, we think that they might also be affecting parasite infection. So that's where we're starting. So Jade collected microplastics from 252 fish. She collected both microplastics and parasites using a special method. So unfortunately, we do have a smaller sample size for this. And she collected them from two main creeks, one exposed to higher pollution and one exposed to lower pollution, to see if we can evaluate correlations between microplastics and parasites. Now, we can't tell directionality, so it could be that microplastics might be making fish more susceptible to parasitic infections, or it might be that fish with parasite infections aren't able to cope with microplastics as well. So we can't tell directionality, but really at this point, any information we can glean about microplastics and parasites is new and important. So this is a graph looking at parasite abundance on the Y and microplastics on the X-axis. And surprisingly, only 18 of the 252 fish had microplastics in their gastrointestinal tract. Now, given the area, there's a lot of urbanization, there's a lot of development, there's a lot of roads. We really expected to see a lot more microplastics. This is very surprising, especially when you look at some other studies that look at microplastics. And other animals, we see pretty high numbers. So one of our concerns is that maybe we're missing something. So our next step is to look at nanoplastics. Nanoplastics are just, as the name suggests, smaller than microplastics. And so our next step is to use what's called Raman spectroscopy to look for nanoplastics. Maybe we're missing them when we dissect the fish because they're too small for us to see with our naked eye. And nanoplastics can actually be even more problematic than microplastics. In humans, they're able to go past the blood-brain barrier. Because they're smaller, they can get more places and they can cause many problems. So we're hoping to use the Raman spectroscopy to find and identify nanoplastics in the gastrointestinal tracts of fish, which we can then start to correlate with our parasite data and maybe get a better picture of what's happening in our system. We're running into a few issues with this method. I think we are probably one of the first to use Raman spectroscopy to find and identify microplastics in museum specimens. So my grad student, Jade, and our collaborator here at KSU are working on to create a protocol for this. They're making great progress. I'm really excited to see what they come up with. But this is our next step. Let's look at nanoplastics. And so to wrap this up, I hope I've convinced you that historical ecology is, a great way to ask questions about long-term long-term changes and how animals are being affected by us over long periods of time. So we're able to collect historical data that otherwise wouldn't be available to us, except, through these sort of creative solutions. And we're able to correlate them with multiple human impacts to get a better idea of how we are affecting our natural environment. And historical ecology is a relatively new field for parasites, but it seems like it's a really powerful way to answer questions that we wouldn't normally be able to ask in natural systems. And while the research I've shown you today in wildlife in general demonstrates declines, and we frame those declines as bad because parasites are important to wildlife and natural ecosystems, historical ecology has been used for many human-related parasites that are showing some alarming results. So, for example, it's been shown that mosquitoes are increasing their range from the historical range, so they're covering more ground and they're being exposed to more people, and so they're carrying these bacterial, viral, and parasitic diseases and exposing new people to them, which is obviously bad. There's been other work showing that human influences like damming or flooding fields is causing some animals like snails to increase, which in areas with parasitic diseases that use snails can increase human disease. So there have been certain places in Africa and South America where this flooding is causing snails to increase, which is increasing incidences of schistosomiasis in people, which is a really bad parasitic disease of people. And so by using these long-term studies, we can get a more complete idea of how humans are affecting the environment, and that might give us some ideas on how to fix it. It might really inform our management decisions. I'm really excited to continue this line of work with the rest of our fish specimens and the other animals I work with as well. But I really wouldn't be able to do this without all the amazing collaborators here at KSU, at the University of Washington, and then my amazing students. I have been very fortunate to have some really, really great undergraduate and graduate students here at KSU, and I'm really thankful that they've enjoyed the work as much as I do. So I'm going to leave you with this video. I do this for all my talks with a botfly coming out of a rodent. It's not bloody, but I wanted to share this with you if the video will play, and I will take any questions you have. Thank you so much.
I'm just fascinated by this video, but if anyone has any questions, sorry, I'm just like watching this parasite. If anyone has any questions, feel free to raise your hand. I'm just thinking, okay, good. I think I can see everybody. Oh, yes. So thank you, Dr. Preisser. Thank you so much for a super interesting talk.
Thank you for having me.
Yeah. Oh, there's a comment in the chat, like, never thought of parasites before. Really cool. Like, I'd certainly never thought of them or the field of historical ecology, but that I think just speaks to my ignorance, but this was fascinating. Mm-hmm. Okay, so there are two questions in the chat. So the first one is somebody who wanted to know if you thought about looking at microscopic parasites as well, like, I'm going to say it wrong, so please correct me, Coccidia?
Coccidia, yeah, close.
Coccidia, sorry, thank you.
So, no, you're fine. So, yes, I have thought about using microparasites, and I've actually proposed for, some other projects looking at, like, toxoplasma. It is difficult to look at microparasites in these museum specimens, but we are doing it in some other, some of our fresh mammal specimens. We're going to start looking at them. So yeah, that's a great point. There's so much to do there. Microparasites cause a lot of problems, a lot of GI issues for people too. So that's a great place to start. I just have a soft spot for the larger worms, so those are primarily my focus, but coccidians would be great.
We have a couple more questions. Did you encounter any resistance from the museums about dissecting their fish specimens? That's a great question.
That is a great question. Usually not, surprisingly. They don't want us to touch type specimens, obviously, any of the type specimens, or if they only have like, a handful of fish from a certain locality or a certain date, we don't want to mess with them because we do somewhat destructive sampling. We take out all the internal organs. sort of shred them while we're looking for parasites. The external fish is mostly okay, so it is still very usable in the museum for the vast majority of people who want to look at museum or want to use museum specimens. So as long as we didn't mess with those more rare or valuable specimens, most curators I've worked with are more than happy to let us use them because they just want their stuff to be used. And so they were more than happy to do so. We had to convince a few, but they came around. They want their stuff used. And so we're careful with it. We return all of the organs back with the inside the jar so someone can use them in the future still. So yeah, not as much resistance as you would think.
Oh, wow. So I think a corollary to that, just wondering if the use of museum specimens means that those specimens are destroyed.
So thankfully, no. Like I mentioned, the external fish is largely the same. It'll have an incision, and we take out the gills from just one side, so that way the gills are intact on the other side for any future studies. The organs are shredded mostly, and we do, mix all the organs back together, so it would make something like stomach content analysis not really feasible. But if a curator wanted us to keep, stomach content separate, we've done that previously, that's definitely doable. But thankfully, no, the specimens are not totally destroyed. In fact, if you were to look at a jar, you wouldn't necessarily be able to tell that we've worked on them.
So another question, what happens if someone eats a fish or snail with parasites?
So it depends. It depends on, one, if that snail is infected. It depends on if it's infected with a parasite that could actually infect people. A lot of parasites, you ingest them accidentally. If they're from wildlife, they may not cause any issues at all or, nothing beyond like a little bit of GI upset. But if you do eat a snail that has a parasite that people can get, depending on, where in the life cycle it is possible you can get infected. So, my suggestion is cook your food. Or, make sure to get food from more reputable sources, maybe stay away from gas station sushi.
I think that's good advice for food in general. So another question, are there any parasite species that are used or could be used as indicator species?
That's a great question. So there have been a lot of propositions to use parasites as indicator species because, they can tell us a lot about what other animals are in the environment if they use those animals as hosts. In practice, not so much. It hasn't really been sort of adopted as a practice in, with management decisions or, outside of academia, but it has been proposed. People seem to agree that it would work. It just hasn't really been done yet.
Great. I'm just looking through to see if anyone's raised their hand. All right. Well, I have a question. I don't really know if it's a question, more just like your thoughts on this. So we had a presentation about the microbiome, and we seem to kind of talk a lot about the microbiome. And so I'm just wondering, like, do you come across that in general, like in your work? Because it sort of seems like these two frameworks used to understand like what's happening in the environment at at a micro level, like kind of the parasitic world and the microbiome, and kind of I was just interested in how those interact.
Yeah, so they are very closely related. There are people who do microbiome and parasite studies. Microbiomes can impact whether parasites are successful in invading or establishing themselves within an animal. They are very closely related, and I have myself have not done microbiome studies, but it is done, and they find that they're both really important and they do interact, and we don't fully understand the extent to which they interact and how much they affect each other, but it does seem like it's a lot more than we think.
One more question. Other than fish, do you know of historical ecology research that uses parasites from other organisms?
Yeah, so actually in my lab, we also work on other animals. And so salamanders, I have a student working on salamanders. I have another student working on snakes. So we'll go to museums, we'll get our snake and salamander specimens and dissect them. My old lab at Texas A&M, where I did my PhD, she was the one that did some of the lice research on specimens. So they would go to museums and they would brush mammal and bird specimens for lice. And surprisingly, the lice are usually there. They don't usually dislodge during the prep process or during, freezer time or anything. And so there are a lot of people using historical ecology field for parasite research. And there's also a lot untapped, though, which is really nice for us.
A couple more questions. How did you become interested in parasites as a field of study?
So I wanted to be a vet. I was convinced I was going to be a vet from, as long as from, the age of 3 when I really discovered animals to when I was going to college. But then in college, I worked at a research lab looking at baboon poop. So I spent hundreds of hours looking at baboon poop under a microscope. And that's when I really gained a great appreciation for parasites. And I decided I didn't just want to treat parasites, I wanted to learn more about them. And so that is what really clicked for me and really made me focus on parasites as a career.
And could human disruption of species habitats also mean that parasites could be more successful? So for instance, in mammals that have begun to scavenge human food and aggregate in human settled areas with greater potential to spread parasites amongst each other.
Definitely. Some species, parasite species, especially those that are what are called generalists, they can infect multiple host species, even across, families or even potentially orders. Those parasites tend to do very well or do better at least in human influenced habitats, especially on species like coyotes and, rats. Species, free living species that are generalists and do really well around human environments. Yeah, they're parasites. Some of them seem to have no problem. And some of my future research, I'm actually looking at parasite traits, using some functional traits to look at what traits are more conducive to parasites being able to survive urban environments. So I'm really excited about that project too. But yeah, there are definitely parasites that can do better around people.
Great. All right, here's another one. Perhaps a bit outside the presentation, but have you given any thought to the hygiene hypothesis, like the idea that the decline in parasites among humans in developed areas may have led to an increase in autoimmune diseases?
So I'm familiar with this. I don't do any work on it myself, but yeah, so there's been some studies where they try to prevent or treat autoimmune issues with parasitic infections, and they've actually seen some success. So I know of some trials using hookworms to help reduce allergies, and it seems to be working. Now, please don't go out and infect yourself with a parasite because you have allergies or some other autoimmune issue. But yeah, because of the immune issue that I talked about earlier, where an immune response to parasite decreases an immune response to something else, there has been a good bit of work looking at how parasites, the absence of parasites might be causing those autoimmune issues to increase, and how maybe using parasites to treat autoimmune issues may be an avenue. I don't do it myself, so I'm not an expert there, but there's definitely work on it.
Oh, wow. Yeah, a couple more comments. Thank you. Very interesting. And I think, yeah, so we have time for one more question if anyone has one. Just scanning through the chat here. Give it one more minute. So, okay, just a thank you in the chat. So I think with that, I'll close the seminar. So I just wanted to say thank you so much, Dr. Preisser. A few more comments, excellent presentation, and thank you. So a lot of thanks. I think everybody really enjoyed it. It was really thought-provoking, and I enjoyed the discussion as well. And so just to say, this is our last One Health seminar of the semester, and I think we're really going out on a high note. So the recording will be up on our YouTube channel in a few days. That's it. Thank you so much for coming.
Thank you so much for having me. I really appreciate it. This was a lot of fun.
Awesome. Thank you.